We're on lesson 14. We just finished with plate 58 on the kidney, and we're going to turn to plate 59. We're going to set up a situation that facilitates filtration. How do we do that? Remember capillary exchange, filtration, and reabsorption. You have a very nice diagram representing the two capillary beds, the blood supply to the nephron. So this is the afferent arterial, the efferent arterial. So blood is coming into the nephron. This is the capillary bed in Bowman's capsule. This is the capillary bed that is around the convoluted tubules. This is the peritubular capillaries. These afferent arterioles have a relatively low resistance, so the pressures coming into these are relatively high. You don't have to remember the number. It's up here, it shows it as 50. In normal capillary beds, the arterial pressure is only about 35. At the arteriolar end, this higher pressure tended to favor filtration. Well, think what happens if you go from 35 to 50. Increased hydrostatic pressure increases the amount of filtration. So the first thing we see is elevated pressures in the afferent capillaries and the glomerular capillaries. So higher pressures. We also talked about fenestrated, that is, capillaries with holes in them. We gave as an example capillaries of the kidney. That increases the permeability. So increased permeability and increased pressure produces tremendous amounts of filtration. So we filter huge volumes, relatively protein-free. We still have the same constraints. We've got capillaries here that are relatively impermeable to proteins. But everything else, glucose, amino acids, fats, hormones, alcohol, anything that is in the blood gets filtered. And it gets filtered without regard to its origin. So you're filtering all of this. Now what you need to do is you need to reabsorb most of it. If you fail to reabsorb that, you're going to urinate it all out. That includes all of those nutrients, all of those important compounds that your body wants to retain. And think about that from a standpoint of how do I deal with the environment that I'm going into. You know, I may be ingesting some bad stuff. I might get exposed to some toxins. And I'm not really sure what I'm going to see in my environment, so let me design a system that filters everything, regardless of whether it's good or bad, but I selectively reabsorb all of those things that I want. So we've set up the model, we're filtering all kinds of stuff. Now what we need to do is we need to reabsorb most of it. And that also includes water. What that means is that what is not reabsorbed gets peed out on the ground and eliminated from our environment, from our body's environment. We have a process to facilitate filtration. Now we need a process that facilitates absorption. And that is in the peritubular capillaries. While the glomerular capillaries were high pressure, peritubular capillaries are low pressure. They're starting at only about 15 millimeters of mercury. They are starting in an environment that favors reabsorption. So in fact, having lower pressures facilitates reabsorption. Now, something else that happens in the peritubular capillaries is secretion. We can modify the urine by secreting compounds into it. Things like urea, penicillin, strangely enough. There are transport pumps for penicillin that move penicillin from the peritubular capillaries into the filtrate. Consequently, dosing regimes, that is, the appropriate dose of penicillin, is actually based on normal functioning kidneys. You can get into a problem if you are treating a renal patient with too much penicillin. They have a compromised ability to secrete, so you start dosing them at a normal level, you can elevate concentrations in penicillin into those individuals and produce neural signs. So, kidney plays an important role in modifying different compounds. They talk about the renal corpuscle. This is kind of an expansion of Bowman's capsule in the glomeruli, and there's a nice diagram down here. So here's a sagittal view. We've kind of cut away. There's Bowman's capsule, so it's lined by cells of the nephron. This is the blood coming in the afferent and leaving the efferent. This is kind of what it looks like in a microscopic external view. And if you look at the internal view, here's these fenestrated capillaries, very permeable, and Bowman's capsule is composed of these cells that have big holes or big spaces. Again, all with the idea of favoring absorption or filtration. So we're filtering a large amount. If we go to plate 60, again, this number is merely for you to hang your hat on. In the human kidney, 120 mils of filter each minute. And they give you some values that if all this fluid, if none of it was reabsorbed, you would essentially urinate your entire volume out in 25 minutes. So clearly reabsorption plays a process. This reabsorption is occurring in the tubular cells. So let's again start right here with our filtrate. It's coming through relatively protein-free. One of the signs of kidney disease is the appearance of protein in the urine. That means you have damaged this interface to the point where you allow proteins pass. Bad news. Okay? Urine should be protein negative. So we're talking relatively protein-free, but what's it got in it? Mostly water, but salts, sodium, chloride, calcium, potassium, glucose, amino acids, vitamins, hormones, other small things. You want to then reabsorb that. So here's the process of reabsorption. You can see that most of the water gets reabsorbed, and most of it gets reabsorbed in the proximal tubule. 70% of the water and material is reabsorbed in the proximal tubules. That is before they go into the loop of Henle. That leaves further modification to be done by the distal tubule and the collecting ducts, which we'll see in a minute. Here's my paratubular capillary. Here's the nephron. And here's the secretory process. And you'll notice that in one of these, penicillin is listed as one of the compounds that is secreted. There's cimetidine, right? You're taking a pharmacological aid for gastric ulcers. There's cimetidine. There are transport. So you have to understand that, again, our pharmacological understanding of how drugs are processed is based primarily on normal functioning kidneys. Let's look at a microscopic view of that and look at more detail of this transport process. This is an enlargement of the cells of the nephron. This is the capillary over here. So these are the peritubular capillaries right here. There's an interstitial space. There is a membrane that is towards the basement or basilar end. So this is sometimes referred to the basilar or basement membrane. And then the membrane that sticks into the lumen of the nephron has all these little fingers. And microscopically, they look kind of like a brush. So we refer to this as the brush border. So cells of the proximal tubule, the nephron, have an increased surface area by the presence of these brushes. And on these brushes or fingers are located transport. Here is a co-transport molecule for sodium and glucose. Yeah, glucose is being filtered in the kidney. It's a small molecule. At the immediate start in Bowman's capsule, the concentration of glucose is identical to what it is in the glomerular capillaries. Remember, the filtration is without regard to concentration. It's strictly driven by hydrostatic pressure. So if you've got 100 milligrams percent of glucose in the glomerular capillaries, that's what it's in Bowman's capsule. Glucose is a precious commodity. In normal individuals, all of that glucose is going to be reabsorbed, and it all gets reabsorbed in the proximal tubule in this process right here. If you exceed the amount of glucose that can be transported by these little transporters, then in fact it's possible to see glucose in the urine. And that is the condition in diabetes. Diabetics have elevated levels of glucose, elevated to the point where they essentially saturate these transporters. And even though these transporters are working as hard as they can and they're moving sodium and glucose from the lumen into the cell, they are not adequate to account for all of the glucose. Glucose appears in the urine. That's one of the signs, one of the clinical signs of a diabetic is excessive urination, and then when you take a sample, it's got glucose in it. So normal urine, protein-free, glucose-free. Sodium and hydrogen are in a co-transport and a co-counter-transport mechanism, which accounts for an acidity of the urine. Well, where does hydrogen ion come from? It comes from the inside. These membranes are virtually impermeable to the charge. So in order for this charge to get out, it must be facilitated through this protein across the membrane. What's the driving pressure? What's going to allow sodium to come in? Well, the first thing is sodium, remember, this is intracellular. The concentration of sodium is very small intracellularly. So initially, there's a tremendous diffusional gradient for sodium to come in this way. So both these processes are passive. The key, though, is not to allow sodium to build up in the cell. To do that, you use this pump right here to pump sodium out of the luminal cell and into the peritubular capillary. This process is an active process. You're pumping ions. We have similar processes for amino acids. Similar transport mechanisms for some of the vitamins. All of these particles are osmotically active. That is, they have a requirement for water. And when you move a solute, when you move a sodium ion, so here this sodium ion is coming into the cell and it's moving this way, water follows. There is no example of active transport of water to our knowledge. That is, all water movement is passive. It's based on an osmotic gradient. So all of the water that gets reabsorbed is passive. Well, This sodium is building up in high concentrations in the interstitial fluid because it's being pumped. It's being forced in this direction. It builds up, it increases here, so what does it do? It diffuses into the peritubular capillaries. Peritubular capillaries, low pressure, so the water follows very easily and accounts for a tremendous reabsorption of water. Calcium, magnesium, hydrogen are also regulated in the proximal tubule, and one of the compounds I forgot was lactate. We talked about lactate way back when being a process of anaerobic metabolism. Lactate is still an important carbohydrate source. So there are transporters for lactic acid in the kidney. You want to reabsorb that carbohydrate moiety and avoid peeing it out on the ground. Plate number 61 talks about measuring filtration, reabsorption, and secretion. I'm not particularly interested in you knowing that for this class. So let's turn our attention then to plate 62. Regulation of GFR. We're going to give the amount that's filtered a name. It is the glomerular filtration rate. We've established a high filtration rate by having high pressures, very porous capillaries, and a very porous Bowman's capsule. So right now we're facilitating filtration, and we have a relatively high GFR, or glomerular filtration rate. How can I modulate that? Well, how do we modulate filtration of capillaries? We modulate blood flow through them by vasoconstriction and vasodilation. The arteriolar tubes, primary site of resistance, primary site of blood flow control in our muscles, in our heart, in our kidney. So figure A shows sympathetic stimulation on the blood supply. This is the afferent and efferent. So there is a separate set of sympathetic controls for the afferent and for the efferent. These are the glomerular capillaries. These are the peritubular capillaries. If you produce a vasoconstriction of the afferent arterioles, that is going to decrease blood flow to those capillaries, and the reduced blood flow and reduced pressure will decrease the driving gradient, and GFR decreases. So in fact, one of the things that happens when you exercise is sympathetic stimulation is increased, blood flow to the kidneys is reduced. GFR, your kidney says, I don't want to spend the energy filtering material. We'll deal with that after exercise. Right now what I want to do is I want to send all of that blood to working muscle. If you constrict only the efferent arterioles, you're moving in the other direction. This, in fact, is going to increase the effective pressures in the glomerular capillaries and increase filtration. What's it going to do downstream? Increased GFR. You'll have decreased pressures down here as well. What's that going to do? That's going to you'll have increased pressures here, decreased pressures here. So you will increase GFR here. You will decrease pressures in the peritubular capillaries, that will facilitate reabsorption. So, filtering more, but you've created now with this vasoconstriction an environment that will facilitate reabsorption of that amount. So in fact, we tend to regulate the amount that's filtered and processed. Figure B shows local response to arterial pressure changes. I've given you an above example of sympathetic nerves, but look here with a local response to arterial pressure. This is without sympathetic nerves. This is a plot of renal blood flow and GFR as a function of arteriolar pressure. In a normal capillary, if I were to increase arteriolar pressure, the amount of filtrate would go up. And we see that right there. There's an increase in blood flow, increase in GFR, amount of filtrate. In a normal capillary, if you keep doing that, it would continue up in this fashion. What we see is a myogenic mechanism in these capillaries that produces, in a response to elevations in arterial pressure, these smooth muscles will contract on their own, the magnitude of which is a function of the arteriolar pressure. So what does that mean? The more the pressure goes up, the more these cells constrict to maintain and regulate the amount of material that's filtered. And that's represented by this graph right here. Amount of flow versus arterial pressure goes up, but the arterioles then start constricting as the pressure goes up, and that brings them back down, so that in fact, over relatively large ranges of arterial pressures, renal blood flow is uninfluenced. That is, your kidney can go on in functioning normally despite changes in arteriolar pressure. Because the capillaries are seeing this blood flow, the amount that gets filtered, or the GFR, is maintained relatively constant. So a wonderful technique for kind of maintaining glomerular filtration rate and renal blood flow over a wide range of pressures. Now we come to the dreaded acid base. I don't know about you. I had great difficulty. My argument was I had no neurons for acid-base knowledge. But in fact, it's so important clinically that I'm going to force you guys to think about it. You already have an idea of CO2 and it's involved. pH is negative the log of hydrogen ion. So anything that elevates hydrogen ion is going to make the individual acidotic, and that is reflected in a decrease in pH. So a decrease in pH is acidotic. We try to maintain the normal pH of our body around 7.4. So this shows you an example of, here's normal plasma. This is the hydrogen ion is denoted by these little colored triangles, and the bicarbonate by the circle. A carbonic acid is the combined. Remember that we have CO2 plus water going to carbonic acid, goes to hydrogen ion and HCO3. I've given you one example of an acidosis in which we elevate CO2, we elevate CO2, that drives the reaction to the right, that elevates hydrogen ion, that makes the individual acidotic. We can also make the individual acidotic by adding in metabolic acids, and the great example is lactic acid. So when your muscle is metabolizing, not only is it producing CO2, which is an acid, but it's also producing metabolic acids like lactic acid. Both act to decrease the pH. We want to regulate pH. Well, how do we regulate that? We have buffers. We talked about one buffer. Hemoglobin is a great buffer. It is a protein buffer. It's found within the red blood cells. But here's our other buffer, bicarbonate, HCO3. A buffer is meant to ameliorate or modify or attenuate changes in pH. A good buffer, if you have a good buffer, that means I can produce acids and I don't see much change in the pH. So here's the first example of changing the pH, holding your breath. You hold your breath, PO2 levels drop, PCO2 goes up. There's a respiratory acidosis. So the individual is becoming acidotic because of the buildup of CO2. You have bicarbonate in there to buffer it. So in fact, instead of getting huge changes in pH, they're buffered. It's modified. Let's go the other way. Let's hyperventilate. Respiratory alkalosis we talked about. In that case, hydrogen ions are eliminated. They're blown out through the lungs, so you end up with way more bicarbonate, right? A lot more bicarbonate. So elevated levels of bicarbonate. Decreased levels of CO2. Lungs and kidney are the key to the buffering system. That's this diagram here. The lungs are the primary site of CO2 regulation, right? Because you can it's a gas. I can get rid of excess CO2 through the lungs. I'm going to affect pH in that fashion. Renal regulation occurs primarily at the level of bicarbonate, reabsorption and retention of that bicarbonate. So how does that occur? Plate 64. Bicarbonate, small molecule, very small. It gets filtered. It gets filtered just like glucose. So in this diagram, this is just odd as all get out. All right, so here's our diagram. This is the proximal tubule. Filtrate coming in. There's the bicarbonate. At Bowman's capsule, the concentration of bicarbonate is the same in Bowman's capsule as it is in the glomerular capillaries. The filtration process is without regard to concentration. But that is an important buffer. If you pee out all that bicarbonate, you are going to become acidotic. So it's critical to reabsorb that bicarbonate. How does that process occur? It occurs by the presence of this compound, this enzyme, carbonic anhydrase. Oh gosh, it keeps showing up. The enzyme that catalyzes the reaction of CO2 and water to form carbonic acid. Well, where does that CO2 come from? That CO2 comes from the CO2 in the filtrate. The same concentration of CO2 that's in Bowman's capsule and in the glomerular capillaries is in Bowman's capsule. So these nephrons are being presented with CO2. This CO2 is drawn into the tubular cells because this enzyme is forcing this reaction. You now have carbonic acid, a hydrogen ion is produced, and a bicarbonate. There are pumps for hydrogen ion. Remember the sodium hydrogen exchange pump that we talked about in terms of reabsorption? Here it is, that hydrogen ion being pumped out, making the environment a little acid. Sodium being moved in. You now have the sodium pumped out actively, right? So this is the same kind of the same diagram, but we've added in bicarbonate. There's a positive charge. It can be coupled with this negative bicarbonate, and you see the appearance of bicarbonate. So this bicarbonate, on the other hand, though, if there are adequate amounts of this combined with a hydrogen ion, form carbonic acid, and the presence of carbonic anhydrase in the brush borders facilitates this production of CO2. So this CO2 can come from the filtrate, or it can come from this production from this bicarbonate. So now, here's this hydrogen ion. Here's the hydrogen ion tied up with the bicarbonate. It combines with this hydrogen ion, which came from inside the cell, which came from this carbonic acid, which came from, wow. So really the process is a little bit convoluted in the sense that, why don't you just have a transporter that moves bicarbonate? It'd be cool, but that's not the way it works. And this system is exquisitely sensitive to the amount of bicarbonate that you have. So if you have high levels of bicarbonate, if you are kind of on the alkalotic side, you're going to swamp the system out, and you won't produce as much CO2, and you won't produce as much hydrogen ion. So you will end up regulating or excreting that excess amount of bicarbonate. If you have very small amounts of bicarbonate, then you will reabsorb essentially all of it. What I want you to know about the distal tubule is transport of ammonia. One of the waste products of protein metabolism is ammonia. Ammonia is relatively toxic. So in fact, the liver does a very nice job of converting ammonia to things like urea. Still, production of ammonia occurs in exercise. Elevated levels of ammonia are associated with poor performance. So wouldn't it be nice to get rid of the ammonia? The distal nephrons serve as a site for the removal of this ammonia. And you'll see down here in this diagram, NH3 ammonia being moved from the distal nephron tubule into the lumen. It combines with hydrogen ion to form ammonium ion, NH4+. this positive charge prevents this ammonium ion from moving back across the membrane. So it makes this membrane, in essence, impermeable to this compound. Well, what happens to this compound? This compound gets eliminated. So in fact, there is nitrogen that appears in your urine. If you exercise a lot, you'll see more of this in the urine. What does it do? It takes a hydrogen ion away. Where did this hydrogen ion come from? It came from H2CO3. You'll see this process right up here. So here's the CO2 molecule coming in, forming carbonic acid because of the carbonic anhydrase. Bicarbonate leaves same way it did in the proximal tubule. It's being coupled to the sodium transport, same thing. But instead of the hydrogen ion combining with bicarbonate, it combines with ammonia. So in the proximal tubule, it's mostly hydrogen ion combining with bicarbonate to regenerate the amount of bicarbonate. Well, by the time we have reached the distal tubule, most of the bicarbonate has been reabsorbed in a natural situation. So now what I have is I have other compounds that can kind of help take out. Ammonia, which is actively secreted, can pick up a hydrogen ion, and also other buffers. Hydrogen phosphates. So we talk about phosphate buffers, and if nothing else in chemistry, you'll come to love, appreciate, and enjoy phosphate buffers. H2PO4, HPO4 are both examples of phosphate buffers. They're in the filtrate, and they can also act to pick up those hydrogen ions. So you see that this hydrogen ion is coming out and can either be picked up by H2PO4, it can be picked up by HPO4, or it can be picked up by ammonia. This hydrogen ion came from this reaction, a reaction which generates bicarbonate. Now this bicarbonate is not tied to a bicarbonate molecule in the filtrate, it's tied to this CO2. That makes this bicarbonate a new bicarbonate. So what we see is in the proximal tubule, virtual reabsorption of all of the bicarbonate that's getting filtered. If I need to adjust the amount of bicarbonate, I can do that in the distal nephron. So if I'm acidotic, if I got lots of CO2, I'm going to generate more HCO3. What's that going to do? That is going to buffer the acidosis. It's going to bring my pH back to normal. If I have low levels of CO2, if I'm a very kind of alkalotic setting, then this process isn't going to go anywhere. I'm not going to generate new bicarbonate. It's alkalotic. I'm probably going to have excess bicarbonate coming through and being excreted. So we talk about pH regulation. We talk about the lungs regulating rapidly, and we talk about the kidneys regulating, but taking much longer period of time. That is, this process is slower. Ventilation is instantaneous or nearly instantaneous. This process takes on the order of minutes to hours to adjust. So again, lungs regulate CO2. They do it very, very rapidly. Kidneys regulate bicarbonate, and they do it a little more slowly. Primary ion in the extracellular environment, sodium. See, a lot of those transport mechanisms are focused on sodium reabsorption. It's a valuable commodity. So let's consider another valuable commodity, one which is the primary intracellular ion. that's plate 65. Here's a schematic diagram of the cell. The large potassium there reflective of high levels of intracellular potassium. This distinction between intracellular and extracellular potassium is important. Recall that it is involved in setting the resting membrane potential. Okay? So that if you alter this relationship, you're going to alter the excitability of neurons and of tissues. So what we see is if the extracellular potassium concentrations drop, you can get a paralysis. If extracellular potassium concentrations increase, you can get super excitation, which may actually produce a fibrillation. Recall that a fibrillation is kind of uncoordinated, uncontrolled contractions of the heart. In order for the heart to function normally, it must have an electronic flow that involves emptying of the atrium, a pause, and then simultaneous contraction and emptying of the ventricles in a very orchestrated, progressive fashion. In fibrillation, all of the different muscle fibers are firing independently. We need to then regulate potassium. Although potassium is abundant in the cell, it's not abundant extracellularly. And this is kind of demonstrated by the difference in the size. And where is this potassium coming from? Potassium intake oftentimes far exceeds the potassium needs. So if you don't want potassium to increase, you must have a mechanism for getting it. So here's a diagram, increased levels of plasma potassium. What's that going to do? So you're confronted with lots of potassium, you're getting hyperexcitable. That increase in potassium is sensed by the adrenal cortex, cells in the adrenal cortex, which secrete a compound called aldosterone. Aldosterone has as its target cell the distal nephron. So in fact, most of our regulation, our tuning of potassium occurs in the distal nephron. At the distal nephron, aldosterone produces an increase in potassium excretion. That's kind of diagrammed by this mechanism right here. Under aldosterone's control, potassium is actively moved into the luminal cell. It builds up in even higher concentrations, producing a concentration gradient that drives the potassium out. So an increase in the distal nephron cell secretion, that is going to then counteract this increase in potassium by decreasing potassium. What's the exchange? Positive ion going out, what's coming back in? Sodium. So aldosterone is important not only in regulating potassium, but also regulating sodium. We actually talked about that in terms of blood volume regulation. Remember, we talked about aldosterone reabsorbing, and we're going to come back and talk about that later. We take a break. What was my time?